Now for our final session of the morning, we have someone who was described yesterday as the DMD mouse expert, the best mouse expert in the world, which is very exciting. DMD mice are used for early stage research. She is Professor Annemieke Artsma-Rus, and she's the Associate Professor of Leiden University's Medical Centre, and she's here to talk to us about preclinical developments.
Thank you very much, and also thank you for inviting me to be at this conference. So I realized when I saw everyone talking about their disclosures, I didn't have a disclosure slide, so I'll just mention it. It's not related to this talk on preclinical research, but just for full transparency. I work on exon skipping normally, and my institute has patents on that, and I'm co-inventor on those patents, so from that I stand to gain money if they're approved. And then I also, like Francesco, Michaela, and Volker, I consult and advise companies, and all the money for that goes to LUMC, so my employer, rather than me. So with that out of the way, I was asked to give a talk about preclinical research and the pipeline, and I'll say, well, why do we need a pipeline? How does the preclinical development work? Some do's and don'ts with concrete examples of how things can go wrong. And then, of course, you want to know what is in the pipeline, what can we expect to come in the future in trials. So why do we need a pipeline? And I think you've heard already this morning, clinical trial drug development takes a long time. It's a lot of work. 20 years ago, we were talking about mouse. We're now talking about clinical trials. It takes time, and it takes all these different steps. And not everything that is at the beginning, that works in the beginning, will make it until the end into the market. So it takes a long time. Not everything will make it. And even those drugs that do make it to the market, well, we've heard some examples in Volker's talk, not all of them are optimal. There's often room for improvement. So what you need new drugs to replace the ones that don't make it to the market. And you also need better drugs for those that do make it into the market. And if by the time something's in the market, you then start to develop that will take you another 15 years. So it's better to have that pipeline full so that every time something fails, something else can take over. So I think that is clear. But how does this preclinical work, how does that happen? And what do we need? So preclinical studies are basic ideas. If we have an idea, we can't just say, okay, now I have this brilliant idea, I'm gonna test it in a patient. That's not ethical. So we need first to see, does this idea work? Does it work in a model system, yes or no? And then if it works, we need to prepare for a clinical trial. What is the best dose? What is the best regimen? How do we administer? Can we do it orally? Do we need to inject it? Can we make the uptake better if we put it in a formulation? And for all of this, you need model system. But bear in mind, the next step is always more complex. So you might hear these press releases or see these scientific publications. They test something in the cell or they test something in the mouse, and it's, hooray, this therapy is around the corner for Duchenne, and everything will be solved. However, the fact that something works in a 2D layer of cells doesn't mean that it will also work in a Duchenne patient. So I think that is important to realize. So preclinical models, there's different types. So we have cell model systems, and these can be primary cells. So we have a muscle biopsy, we isolate cells from that, we culture them in a dish. We can do that for a small amount of time, but at some point they stop proliferating because, well, we don't have the eternal life. At some point regeneration stops. So the primary cells, we can use them, but only for a while. They've discovered a way to immortalize muscle cells, so then they can grow almost indefinitely, and we can use that to study them, but of course they're not the same as muscle cells. We adapted them a little bit to make them immortal. And there's now also muscle bundles. So in a culture dish, these cells, well, they look like muscle fibers, and this is also called muscle on a chip. So we have these culture systems that can be used, and usually these are human-derived. So we can test our human-specific genetic approaches on these human-derived cells. Then we have animal models, and there is a plethora. There's mice, dogs, rats, worms, fish, flies, pigs. There is a whole lot. However, these models, usually, they have their mouse dystrophin gene, their fly dystrophin gene. It's not the same. So if you do genetic therapies, often you cannot use what you would use in a human. There's some exceptions for the mouse. Generally speaking, there is a translation that you have to make. This works for the mouse, and now we need to adapt it for the human. So, introducing you to the MDX mouse, Emily mentioned it already yesterday. The MDX mouse is the most used model for Duchenne. And it's a spontaneous model, so we didn't make it. The mouse had a spontaneous mutation in its mouse dystrophin gene, cannot make dystrophin. And it has pathology, so it has muscle breakdown, it has muscle damage. In the lab, they do really well, but if you look at them, you can see they fatigue more easily, they cannot run as far, they are less strong. So in the wild, probably they wouldn't do very well because, well, it's the slowest mouse that is caught and eaten. In the lab, they don't have this problem, so they do quite well. But mice have a really good regenerative capacity, and that means that they cope with not having dystrophin really well. So they have quite good muscle quality. And the problem with that is that all the therapeutic approaches that we develop, they rely on the muscle tissue that is there. And that means that it will always work better in the mouse because there is a lot more muscle tissue and good quality muscle tissue there than there is in a patient. So that's just something we have to bear in mind. Things will just work better in the mouse, sadly, than in the human. Mice also have a very quick metabolism. And that means that if something is safe in the mouse, that is by no means a guarantee that it will also be safe in the humans because we just don't have that fast metabolism and we cannot cope with certain things. So you need to know, with all your model systems, you need to know the limitations and you need to ask the model the right questions. And what you also need, you need to understand the natural history of the model. Like you need to understand the natural history of Duchenne, because then you can plan your trials properly. Same for mouse models. And what's interesting is that, well, this MDX mouse has been around for very long, I think over 30 years, but no one has really looked at it systematically in a large natural history study. And it's currently ongoing. Ongoing at LUMC in my group in a collaboration with Annemarie De Luca from Bari with sponsoring from Duchenne UK. So it's ongoing, but it's a lot of work. So, well, the work is done, the analysis is ongoing and pending. So you now have some idea of why we need a pipeline and how these preclinical development, how this works. I'll give you some examples just so that you know how things can sometimes be misconstrued and how things can not work optimally. And so some do's and don'ts. So you need, I said already, you need to ask your model the right question. You need to understand the model and then ask the right question. And just one example. So we have a worm model, the C. elegans. It's a really small worm, so that's easy because if you want to do high throughput screening, you can test a lot of different drugs on these little tiny worms. But this worm doesn't have an immune system. So if you want to study the impact of inflammation on not having dystrophin and the pathology, this worm is not going to tell you that because it doesn't have the immune system. Also, if you do your drug screening, you will not find anti-inflammatory drugs because, well, the model doesn't have an immune system, so this component you will not find. Very interesting. So we know that the steroids, we always say they're doing their job because they are anti-inflammatory. And that is part of the story. But there's more, because if you give this worm steroids, the worm without dystrophin, it gets better. And it doesn't have an immune system, so it must be something else that's happening. So we know in humans probably other things are happening as well. That as a side note. Very important to use control animals, so animals that do have dystrophin, so that you can see what is the difference. So if I don't have dystrophin, this model has, well, it cannot crawl as quick as or as well as the worm with dystrophin. It's less strong than the mouse without dystrophin, etc. Really important. So these models, they have their own way of coping with not having dystrophin. And some of the models do weird things that aren't happening in humans. So there's a mouse model, not the MDX, but a slightly different model, that makes calcification in the muscle. So that's not what's happening in humans generally. So then if you try to treat that calcification, well, that is great for that mouse model, but for the humans, it's not really something that would help because we don't have that issue. So try to not treat the model, but try to treat the disease. And again, if you look into publications, sometimes people do this. And don't skip steps, and I'll show examples of that. So here what we see is a mouse that's treated. So we have MDX in orange, and then the treated MDX mouse in blue, and we see the diaphragm function, so respiratory function. You see the treated mouse does better. And you could even say, well, it goes from 5 to 8, so it's 60% better. And if you say it like that, it sounds, well, quite something. Now I'm going to show you the normal mouse. And suddenly, the difference is a lot less prominent, because the normal mouse is at 100. So yeah, the treated one does better, but you can see the deficit with normal is still huge. So you really need to have this wild-type mouse in order to see, is this treatment really relevant, or are we just looking into slight variations and noise? So don't blame the mouse for everything that goes wrong. So there's so many things that work in the mouse. Why doesn't it work in clinical trials? Well, sometimes that is because, well, we aren't mouse and it didn't predict. But often there's also something else happening, and that is that drug developers sometimes just want to skip steps. And they say, well, we've shown proof of concept. We have this idea, it works in the mouse, and now well, we could do all these preclinical studies to optimize the dose and the regimen and the formulation, etc. But there's an urgent need for therapies. And well, you will all agree, there is an urgent need for therapies. But if you skip these steps and then say, we're immediately going to go into the trials, what will happen is that these trials are not optimal. And we've seen examples of that in the past, where there was a drug that was used at a high dose, and then that resulted in side effects. So I think out of the 20 patients in the trial, only 1 completed it. Everyone else stopped because the side effects were so severe. And then they said afterwards, maybe we could have used a lower dose. Well, we didn't test it in the mouse, so we don't know whether that would have worked. We just tested what worked in the mouse. Well, you will argue that that's not optimal, and that's a wasted opportunity. And then you can say, well, things go quicker if we skip steps. Well, not really. The other way around can also happen, where something doesn't work, and they say, well, but maybe we should have used a different regimen. Maybe not every week was not enough. Maybe we should have done it daily. So really, these extra studies in the mouse, they are crucial. You need to get all the information that you need to optimally plan your trial. And of course, if you skip steps, you can't really blame the mice for that. You can only blame the people who developed this for that. So blame the humans, not the mouse. So then, of course, what you're interested in, what is currently in the pipeline? And there is a lot in the pipeline, and I have only 35 minutes, so I won't be able to go over everything. There's ways to improve exon-skipping drugs and gene therapy, but we already had Volker and Francesco talking about that, so I'm not going to talk about this. I will talk about gene editing and stem cells, and then also there's therapies that target pathology, and again, we had Michaela give a presentation about some of these approaches. There is a lot happening, trying to improve the mitochondria, so the small organelles in our cells that make energy, improve energy metabolism, antifibrotics. There's a lot happening. I will give an example of drug repurposing. And finally, if there's time, I will give one example, something that Volker mentioned as well this morning, confirmation bias. As researchers, we're also human. We believe that our idea works. And then sometimes we're just so obsessed with that we fail to see red flags that something might not work. And I have a published example of that. We'll start with cell therapy. And cell therapy is something that people thought about before we even knew that Duchenne was caused by lack of dystrophin. Because the idea is really simple. There's muscle damage. What if we isolate the muscle stem cells, we expand them in a culture dish, and then we transplant them into a patient? So we isolate the healthy cells, we transplant them in a patient. whatever protein is missing, these new cells will start to produce. And also, their stem cells, what they do, they repair the muscle. So we'll get 2 for the price of 1. And what I show here is a muscle fiber from a mouse. You see these little bulges on top. Those are the muscle stem cells. So you need to isolate these. And in theory, the stem cell treatment, it makes a lot of sense. However, the reality is that it doesn't work. And it doesn't work because if you inject the stem cells in the bloodstream, they don't go to muscle. If you then say, well, let's inject them into the muscle and hope that they migrate there, no, they don't. They're lazy, they will stick and stay where they are. So this is very, very inefficient. So you introduce maybe hundreds of thousands or millions of stem cells, only a few here and there will actually go into a muscle. And then that muscle will be dystrophic. There is no dystrophin, there's inflammation, there's fat infiltration, there's fibrosis, and all these processes will give signals to the stem cells, to the muscle stem cells of the patient, but also these transplanted cells. So what will happen, these transplanted cells will not start to make muscle, they will become part of the fibrosis, they will become part of the fat tissue that also the body's own cells are doing. So sadly, at the moment, there's just zero evidence that this works, and that's because it's not possible to deliver. If you want stem cell treatment and you're willing to pay for it, you can get it. There's promises. You go online, you can get it in Russia, you can get it in India, you can get it in many places if you pay for it. Of course, so there's no evidence that it works. You can say, well, it might not harm. Well, you don't know what they treat you with, so it might actually do harm, and it will definitely harm your wallet. So if you have to spend 10,000, 50,000 euros, I think there's much better ways to spend it than trying this. So then gene editing, and you probably know about gene editing as well because it's in the news, it's hyped. What is it? So gene editing is a scissor that cuts in the DNA and a GPS that tells the scissor where to cut. And why would you want this? And well, I had anticipated that Francesco would have explained exon skipping, but he didn't. So I will explain exon skipping, and then I'll explain why gene editing would be an advantage. So what exon skipping does, it tries to restore the gene code for dystrophin. So what we see here, we see the gene code. So these are these exons, these blocks. And what we see in this example, 52 is missing. And we see 51 doesn't fit to 53. So the gene code is disrupted. And when the cell starts to translate this into protein, it works until 51. And then in 53, the code becomes unreadable, translation stops. And that means only the beginning of the protein is made, and only the beginning of the protein is not functional because dystrophin provides a link between the skeleton of the muscle fiber and the connective tissue surrounding the muscle fiber. So this is unreadable. What you can see is 50 would fit to 53. And that is what we see in Becker muscular dystrophy patients. So this would fit. The code can be translated. There's a little bit missing, but the part that binds to the skeleton and the part that binds to the connective tissue, these parts are there. So it's a shorter dystrophin, it's partially functional, and therefore Becker patients have a slower disease progression than Duchenne. So what we try with exon skipping, we try on RNA. So we have the DNA. If protein needs to be made, there is a temporary copy called RNA, and that is then translated. So with exon skipping, we try to manipulate this. And RNA is there for a little while. So that means that you need to treat with exon skipping drugs, you need to treat every week or every month, depending on which approach you use. But it's repeated treatment, which of course is a burden. If you could edit the mistake on DNA level, so here, removing exon 51, then every time an RNA is made, it would be a readable code, so then every time a protein can be made. And this is one cell, so of course it doesn't work if you do this in just one muscle fiber, that's not going to be enough. You need to correct this in all the different muscle fibers to really have an impact. But you would have to do it once, and then you would get this effect permanently. So in theory, again, it sounds great, and it's hyped, especially when the gene editing, the CRISPR-Cas9 system, got the Nobel Prize for chemistry. Chemistry, not medicine, because there is not that many drugs that work yet. And it has potential for Duchenne, and you can do it in cells, you can do it in mice. We know this. We can restore dystrophin with gene editing. But the delivery is a lot more challenging than if we do the microdystrophin. And also, there's gene editing drugs approved now. And so you can say, well, if it's approved for this blood disease and this liver disease, why isn't it also then doable for Duchenne? Because muscle is, again, so difficult to reach. So what is approved is ex vivo. So you have a blood disease, you take blood stem cells, bone marrow stem cells from the patient, you edit them in the culture dish, you do a quality control to make sure that nothing weird happens, and then you transplant the cells back. So that is approved. It's also ongoing for a liver disease where things seem to work well, but what do you do with the liver? You can give a lipid nanoparticle, so a fat nanoparticle, where you package the RNA for the Cas9, for the scissor enzyme. So that means that scissor is there maybe a week at most. So it's there very shortly. So it can do its job and then it's done. If you deliver it for Duchenne, which the only vehicle we have at the moment is AAV, that means that it will be there for much longer, for years and years and years. And you can say, well, that's good, because if it doesn't do its job at first, it may do it after a week or after a month. But it also means that it has a week, month, years to do what it's not supposed to be doing. And we know that that happens as well, to cut where it shouldn't cut and to cause DNA damage. And that is currently a big challenge. So there's people that mention CRISPR cure. For Duchenne, CRISPR will not be a cure for two reasons. The first one, what will happen is the patients will start producing a Becker-like dystrophin. That is not a normal dystrophin, so it's unrealistic to expect that this will cure someone. Also, you will treat at a certain time point, and all the damage that has accumulated sadly will not be undone. So if you have a fully dystrophic muscle and suddenly that muscle can produce dystrophin, that muscle that isn't muscle anymore will not suddenly become muscle just because now it can make dystrophin. Dystrophin doesn't make a muscle, muscle tissue makes muscle. So that is also just to make you aware, it's not a cure, even though people call it CRISPR cure. So there's things to optimize for Duchenne. The delivery, off-target edits, how sure are we that it only does what it's supposed to do? And then something that we now see is this Cas9 is an enzyme, the scissor enzyme comes from a bacterium. We have exposure to bacteria all the time, so a lot of people have antibodies against this Cas9, and that might be a problem. If you then start to produce it, it's not just that it may prevent the action, it may also cause an autoimmune response where every cell that produces this is now attacked by the immune system, and of course you don't want that to happen to your heart and muscle tissue. So there's things that we need to optimize. So then drug repurposing, so what is this? So we have a drug that is approved for a specific disease, and it targets fibrosis or it targets inflammation. And we say, well, that's a pathological feature that also happens in Duchenne. Can we use that drug that's approved for instance, pulmonary hypertension? Can we also use it in Duchenne? Because if it's approved for something, there's a lot of data already. It should be easier and quicker. So yes, this of course is a great idea. And often then people will start again in the MDX mouse and show, look, it works. But there's certain things to bear in mind. The dose that is used. So lots of times when these things work in the MDX mouse, if you look at the dose that is used, and then you see what dose is used in the humans, you see, well, but now they use 10 times, 50 times, 100 times more. Yeah, the MDX mouse will tolerate this. Humans will not. So then, yes, you need, with a high dose, you can achieve great things in the mouse, but you will never be able to do this in a human. Also, safety is an important consideration. The fact that it's safe for one disease doesn't mean that it will also be safe in another disease, because there's other pathological features. For instance, with Duchenne, there's a lot of muscle breakdown. Well, that poses some stress on the kidney. So the fact that it is safe for the kidney in a disease where there's no kidney involvement whatsoever doesn't mean that it will also be safe in Duchenne. Duchenne patients are using corticosteroids. If it's approved for a disease where no one uses corticosteroids, you will need to check, is there an interaction? Because maybe combining this leads to safety concerns. So you need to test this. So you really need to bear that in mind. And of course, mice are not humans. And I have one example of how mice are not humans and how repurposing doesn't always work out. So this is sildenafil, and you all know sildenafil. Sildenafil is the blue pill from Pfizer, Viagra. So what people did, they did a study in MDX mice, and MDX mice, they develop later in life heart problems, just like Duchenne patients. And what they could show, if they treated with Viagra, the heart pathology, the fibrosis in the heart, went away. So it's not that it prevented it, it was there and it went away, which is, of course, really amazing. But again, mice are not humans. And obviously, this was something that was picked up by the media as well, because it was, well, everyone liked the headings, Viagra to treat muscular dystrophy. So what happened then? Two things happened. First of all, some researchers in the US said, well, if this works, then we need to test this in a clinical trial. Properly test it in Duchenne and Becker patients. What happens if we treat them with Viagra? Will this indeed reduce the heart problems? But then, in parallel, some parents said, well, we can get Viagra, we buy it on the black market, or the father said, well, I'll pretend I have erectile dysfunction, I get it, I give it to my sons. And that happened in parallel with the trial. Now, the trial showed it didn't have a therapeutic effect in patients. And the heading doesn't say, but if you read into the paper, actually what it did, it made the heart condition in Duchenne patients worse. So treating was making things worse. And in parallel, these families had been feeding their children Viagra and then realized, well, actually, well, yeah, it worked in mouse, but it didn't in humans. So again, like, really, there's a reason we do these trials. It's really important to sometimes be patient because you can make things worse. Then the last example I mentioned already is confirmation bias. As researchers, we are human, we have ideas, and of course we like to think that we are clever and that we like to think that we are right. So we will look for ways to confirm that we're right, even though science tells us that we need to look for ways to show that we are wrong. So this is an example, and this is published about an idea to use stem cell treatment for Duchenne. So it's quite convoluted, but I'll explain. So what do? They say, well, if you do stem cell treatment, one problem that I didn't even mention is if I would transplant cells from a healthy father into a son with Duchenne, there's an immune response because the cells from the father will be recognized as straight. So there will be an immune response. So this is bad, they said. So what do? They isolate muscle stem cells from the father and from the son. Then they give them a color. So the father, the cells, they add a gene to make them red. The cells from the son, they add a gene to make them green. And then they fuse them. So they fuse the cells from the father and the son, and in fluorescent, red plus green makes yellow. So they isolate the yellow cells, the cells that have characteristics from both the father. They can make dystrophin, and the son, they will be recognized as the own cells, and then they transplant them. But we know already you cannot transplant these cells because if you transplant them in the bloodstream, they're not going to go to muscle. If you transplant them into the muscle, they will just stay where they are. The solution, the researchers found, is what if we inject it into the pelvic bone? It will sit there, and then regularly these cells will move out and go to muscle. That's their hypothesis. And then, of course, the patient will get better. So this, I mean, I'm not making this up. This is actually in the paper. So this is what they did. And of course, they couldn't do this without preclinical studies. So they tested this in the MDX mouse. If you read the paper, they didn't use any control mice. And they say, we get 15% dystrophin. Those of you who've been around for longer know that 15% dystrophin can mean different things. What they meant is 15% of fibers make some dystrophin, and some can be a very flexible term, of course. So they make really, really very little dystrophin to the point where you can think, is this background or is this a treatment effect? And then they say the treatment improves the heart. So again, there's no healthy mice, so all we have is baseline, so before the treatment, and then with time they see that the heart function goes down. So these mice are not treated, so you see with time the heart function goes down. Then these mice receive the low dose, baseline, and again you see with time the heart function go down. Then they say these mice are treated with a high dose, and look, the heart function stays the same. Well, yeah, they're not lying. But if you're clever, you can see, well, but this baseline bar, is lower than these two baseline bars. So is there a treatment effect, or just by chance, was their baseline lower? Like, this is not a treatment effect because nothing goes down, it's just that this was lower. But based on this, they said, look, this works. So now we're going to move into trials. 3 patients treated, 2 6-year-olds, 1 15-year-old. They fused these cells from the father and the sons. They did a biopsy in both. They transplanted it into the pelvic bone under anesthesia. They did 6-month follow-up, and they said, look, the ambulant patient, the 2 6-year-olds, they improve on the 6-minute walk test and on the North Star. Well, what do boys who are not treated, the most 6-year-olds who are not treated, will improve on the North Star and the 6-minute walk test? So you don't have to treat them to get that effect. But since they were so much looking for the ways that their approach was working, they said, this is because we've treated them. Then they said, this is because we've treated them and they are making dystrophin. And so then you would say, well, show us a biopsy. Show in a biopsy that there is dystrophin. They said, no, it's unethical to take a biopsy. Even though they took one here at the beginning and they did the whole anesthesia for the cell infusion, they said, no, it's unethical to take a biopsy. But they conclude, based on these 3 patients, this works. And again, so I don't want to badmouth these authors and these researchers. This is very, like, researchers, we like to be right. So it's human, but it does tell you that you should not believe everything that you read, and you should be aware that also these researchers, they like to believe that they are right. So have to be a little bit more critical, and there's a clear difference between a clinical trial that was shown by Michaela and by Volker and by Francesco, where you have hundreds of patients, where you have a placebo group, versus 3 patients who are treated without any controls. So take-home messages, learning objectives. You need preclinical research to keep feeding the clinical pipeline. You need model systems. They are not perfect, they are model systems. So you need to ask the right question to the right model. And preclinical research should prepare for clinical trials. So that's also something, if you're considering to be in a clinical trial, to ask, so what did they do? Why do they do this dose? What studies did they do to make sure that it's this dose and not anything else, etc. Skipping these steps will lead to suboptimal clinical trials, and I think that's something that we've learned. And also there's now the TreatMD Advisory Committee for Therapeutics, where companies and academics who want to do trials, they can come and get feedback from a multidisciplinary team. And then, of course, if we see these things happen, you need to do more preclinical work, and that's very often one of the feedbacks that we provide. And be aware that not all approaches that are in preclinical research will make it into the clinical trial stage. So I think Michaela also said, like, you can wait until the gene editing trial is there, but it might not be there next year, it might not be there in 5 years, it might never be there. So you really have to balance this. There's an uncertainty we don't know. Preclinical research, also there, things fail. So with that, I want to thank you. If there's time for questions, I'm happy to answer questions, and I'll be here all day, so you can also approach me during the breaks.
Question if there is one, and it looks like somebody at the back
there. Hi, in terms of the pipeline, is there any progress in the delivery of full-length dystrophin? For example, the sort of lentivirus work, myosana and so on?
Yeah, so there's work on trying to deliver full-length dystrophin. with different viruses, but there's a reason that everyone uses the AAV to deliver and to make things fit in the AAV, and that's the AAV is so far the only thing that efficiently delivers to muscle. This way where they try to, say, make a combination of AAV and lentivirus to give the lentivirus a way to deliver to muscle. So far, that's in very early stage, and that needs a lot of optimization. And I think one thing also to consider, if you want to deliver full-length dystrophin, and this is something that we didn't discuss yet here, but what we see with the microdystrophin, if we deliver a domain in the microdystrophin that is not present in the patient, so there's a deletion that in the patient that involves that domain, like the beginning of the dystrophin. If we then give a microdystrophin that has that domain, that can lead to autoimmunity. So then the cells start to attack the cells that the immune system starts to attack the muscles and the heart that makes this foreign domain. If we deliver full-length dystrophin, for sure we will deliver domains that patients don't make, because most patients will have deletions, so we're delivering parts that they've never seen. And then the question is, will each of these parts lead to an immune system? We don't know, but there is this risk. So there's also the question like, is this really the solution? Well, it might be, but we have to bear in mind, we know already that there's parts of dystrophin that will lead to an immune response. So for those patients, it will not be a solution. So there's work ongoing, but it's good to be aware that it can also go wrong, and it might not be a realistic solution.
Okay. We'll take one more, there is one, otherwise we'll move on. No, I think that's good.
Okay, thank you.
Thank you, Professor Artsma-Rus. Now we